Well, thank you very much. Hello, neighbors. I'm just from a few blocks away. So today, I'm going to tell you a little bit about what we do, mostly about what we think about this, this unknown area of biology. I like to start by saying that it's a great pleasure. It has been such a great pleasure to be living in St. Louis. Right? This is a great city. We have that arch there, and I actually take advantage of that art, and we'll call that the gateway to the human epigenome. Toward the end, I'll tell you why we say so. So Alex told me to talk about charting the unknowns. I'm from the genetics department at Washington University. Right? Genetics is a very, very interesting topic, especially in the past 15 years, we have gained so much knowledge about our own genome, to the degree that we feel that, we need to know our own genetic component in order to have a healthy and interesting life, right? So what is human genome? This is our genome, right? This is probably one of the biggest now known unknowns out there, right? Every single cell in our genome has this long string of letters that's called the blueprint of humanity, right? If you print our genome into book volumes, sorry, I'm gonna have to, how do I go back? Huh? Okay, so if we print our genome into books, this is how much volume it takes, right? When I was a little kid, I loved to read, right? So this is probably one of the reasons that I was attracted to genome science. I want to understand what this book is about, right? This is so-called a human reference genome. Now, everyone has a different set of books because, we are separated by a certain level of genetic variation, and that variation is supposed to underlie the phenotypic differences between individuals, right? So this is sort of a big known unknown. I mean, we have such a big genome. We know it's there, right? But the biggest challenge is how to understand this genome. Now, I'm going to pose a different question. That is, a big unknown that we're going to chart is to how to read this genome, right? Not about how to just figure out the differences between different genomes, but think about this question, right? Every single cell in our body essentially shares the same genetic information. It's the same set of genome that we got from our parents, right? But our fibroblast cells look very different from neuronal cells. How different cells with the same genetic component, right, the same genome, look very different, right? So this, to us, is a big unknown that we would like to chart. So the question that I want to propose is this. We are our genes, right? We're what our gene tells us to do, right? But there's something else. There's something on top of genetics. There's something on top of the genes, and that's the big unknown that we want to chart. I want to begin by giving you some interesting examples. Now, on top of genes, this is what we call epigenetics, right? So the first example in epigenetics that I want to introduce to you are these two lovely mice, right? Look at the mice, how lovely they are. Don't you want to cuddle them, right? Especially the one on the left, right? It's chubby, it's golden, you just want to hold on to it, right? So these two mice clearly look very, very different. Unfortunately, the one on the left, it looks much prettier to me, but it actually has a, kind of an unfortunate fate. It's unhealthy, right? It has high risk of cancer, a high risk of diabetes and obesity, and it has a reduced lifespan, okay? The mouse on the right is, by and large, a normal mouse. Now, what I'm going to tell you is, these two animals, these two mice, right, they have identical genomes. Their genetic information, right, from the first base in their genome to the last base of their genome, right, are identical. They're more identical than identical twins, right? Not only that, these two animals, they live in the same condition, they live in the same cage, they eat the same food, everything about their life are identical. Now, I just told you, right, genes are what we are, right? We are determined by the genetic component of our genome. But this clearly violates that rules, right? These two mice, they look very, very different. They have very, very different fate, right? Yet, their genomes are identical from the first base to the last base. So where does the difference come from? Turns out, the only difference between these two mice, right, is the diet of their mothers when their mother were pregnant. Right? Turns out, the mouse on the left, right, when his mother was pregnant with him, right, she just ate normal food, a normal child, the lab child that we gave lab animals. Right? The animal on the right, the mouse on the right, in addition to normal food, his mother also took vitamin supplements. Right? I'm not doing any commercials here, but but she took vitamin supplement like folic acid during her pregnancy. Right? So as a result, everything else about these two animals are identical, but the only difference is their mother's diet, but as a result, they have very, very different phenotype, they have very, very different life. So this is a perfect example where I say, we're not what we are, we're what our mother eats, right? Give us great reason to blame our parents for the things that we do in our life, right? So, as a scientist, we're interested in understanding the molecular mechanism of this. And it turns out this is the molecular mechanism of these two animals, right? Their genomes are identical. Turns out the causal gene is called agouti. That's the black boxes there. Far, far away from that gene, about 100 kb, this is a very long away from that gene, there is a genetic element called transposable element. This is something like a virus insertion in this mouse's genome, right? So that element is able to drive the overexpression of that agouti gene at the time and in the tissue type where this gene should not be expressed, right? All of this resulted in that bad phenotype, fat and sort of diabetic and cancer-prone, all this bad phenotype, right? When the mother was pregnant and when she takes folic acid as a vitamin supplement, folic acid provides something called methyl donor, right? So this chemical can modify that part of the genome, not the sequences, not the genetic sequences, ACGT, but the chemical modification on top of that viral element insertion, right? And that modification is called DNA methylation. That methylation can silence that transposable element or viral element, and then it no longer drives the overexpression of that gene, so the phenotype, the gene expression return to normal, and then the phenotype return to normal, right? So again, this is a epigenetic modification, in this case, a DNA methylation on top of our genome that governs when and how our genomes are expressed. And that directly resulted in the very different phenotype we experience. Now, this is what we're interested in understanding. That is, given the same genome, right, what kind of modifications happen on this genome that's going to relate in our health and in disease. Okay, so this is one classic example of epigenetics, and I'm going to come back to that example a little bit later. I'll give you another example just to make, these are the sort of the fun things that we think about and then work on in the lab, right? So what I'm showing you here are the honeybees, right? Can you tell me where's the queen bee? Where's the queen bee? Just look at those lovely pictures, lovely, bees. The queen bee is right in the middle, right? That's where I put it there for you, right? The queen bee is right there. Now, the queen bee looks a little bit different from the worker bees, right? It's a little bit bigger. But more importantly, the queen bee has a very, very different life from the worker bees. It lives longer. It never leaves its hive. Right? It lives much longer than the workers. It lives like several years, when the workers only live a few months. The queen bee does nothing but give birth to babies. So I don't know if that's a good life or a bad life, but that's a very different life, right? So she gets to live much longer. What I'm going to tell you is the queen was not born a queen, right? In fact, when they were larvae, queen bees and worker bees are identical. They're genetically identical. The individuals are all identical, right? A few of them are sort of randomly chosen, and they were fed a different food, right? The larvae that got fed royal jelly got to become the queen, right? And the rest of the bees got, just normal food that we eat, and they become the workers, right? So again, genetically, they're identical. It's not the genes, right? But by having different food, right, different environment, essentially, they have very, very different fate. And this is what we are interested in understanding as epigenetics. It's not about the genes, it's how the genes are expressed that determine life, health, disease, and so on. Okay, so this is a big unknown that we want to chart. So in this case, we also linked this particular phenotype to a molecular mechanism called DNA methylation, very similar to the mouse example that I just described. Turns out the worker bee's genomes have a higher methylation level than the queen bee's genome, to the degree that if you go ahead and then take a worker bee and you use an experimental strategy to inhibit the DNA methyltransferase, which is responsible for methylating the genome, okay, you can actually turn a worker bee into a queen bee-like feature, right? So if you inhibit DNA methylation on in a worker bee, right, a worker bee will grow an ovary, right? So again, this has nothing to do with their genetic difference, because workers and queens, genetically they're identical, right? But because they eat different food, which resulted in different level of DNA methylation across their genome, they ended up having very, very different phenotype, okay? So this is another classic example in the field. I'm going to bring a third example. Now, this one is completely unknown, but I think it has epigenetics written all over it. This is a documentary that I watched. I took my kids to the Science Center about 3 years ago, during Thanksgiving break, right? So we watched this documentary about monarch butterflies. This is an amazing creature, right? So the documentary is about finding where they spend the winter in Mexico, right? This is one of the migration animals, right? So it turns out that they spend the winter in Mexico, and they spend the spring and summer in the continental North America, right? So the documentary is about how scientists were able to trace the migration path and found the secret location in Mexico, where this butterflies spend their winter. Now, so that's cool, but many animals migrate. What's special about these monarch butterflies? Now, they do this in 3 to 5 generations. They do this in 3 to 5 generations. In other words, the butterfly who left Mexico, they will only fly to somewhere in Texas, and then they stop there. They give birth to their babies and daughters, and then these butterflies will just die there, okay? And those daughters will continue flying north to, say, Wisconsin, right? And then they're gonna stop there, and then they're gonna give birth to their babies and their daughters, and continue migrating north and then all the way to Canada, okay? And then they give birth to the next generation. Now, it's that generation, the generation that was born in Canada, somehow knows to fly all the way back to Mexico. Again, these generations, they have identical genome. Genetically, they're identical. But they have very, very different life fate. They have very, very different behavior. Now, I don't know. I mean, for me, this has epigenetics written all over it. We don't know yet, so this is something that I would love to work on one day if I get to do a sabbatical, right? So I'm working on it. So all of these are the interesting phenomena that we call epigenetics that attracted me to this field. But you're gonna ask, well, does it happen to humans? Does it happen to us? In fact, it does. It happens to us all the time, every day. Everybody is a walking bag of epigenetics, right? So as I mentioned at the very beginning, the fundamental question that we address in the lab is this, right? Every cell in our body essentially share the same genome, but why the cells look very different, right? What determines the cell fate, and how does this cell fate evolve, right? So these are the questions that we are interested in understanding to investigate in our own labs. Now, epigenetics was initially described as used to describe development, right? So how one cell can grow into a multicellular organism. Every cell now has the same genetic information by and large. There's a little bit changes here and there, but by and large they have the same genome, but they have very, very different epigenome. They have very different epigenetic regulation that resulted in this phenotypic differences, right? So this is the sort of the fundamental question that we want to understand: how this one genome, right? One genome is able to encode a wide variety of different cell types, which we call a wide variety of epigenomes. Okay. Now, again, bring back to the concept, same genome. By having different epigenome on top of the same genome, we ended up having different phenotypes, as individuals and as individual cell types. So what is epigenome, right? So these are the chemical modifications happening on top of our genome. So this is just a sort of a collection of the type of modifications that we know of. Again, we know these are the modifications. I talked about DNA methylation, a little chemical modification on top of our genome. There's also histone modification and some, many other things. So we know these modifications. The unknown is how do these modifications work, right? Where do they happen? What directs them to happen on different parts of the genome, and how they dictate different part of the genome to express at different time in different cell types, right? So these are still largely unknown. But one of the important tasks in our field is to chart this unknown, right? Figure out what are the DNA, where are the DNA methylation that's happening in our genome across different cell types? Does it happen the same way between you and me? Does it happen in the same way or different ways between human and other species? So these are the fundamental questions that we're sort of, the unknowns that we're charting. Okay. Epigenetics has been used to describe development, and one of the most interesting analogy people make is this epigenetic landscape. To understand this, you imagine you put a little ball on top of a hill. When this little ball has a lot of so-called potential, now we call this epigenetic potential. And then we let this little ball roll down this hill. And at many branch point, this ball will have to make a decision: do I go left or right, right? So during the course of development, this is essentially how a cell make a decision. Do I commit to the neuronal lineage or do I commit to the blood lineage, right? To the point when this little ball get to the bottom of the mountain, and now we call them lineage or lineage terminally differentiated. So these cells no longer become other cells, but when they are up on the hill, those little cells have the ability to become many, many different cells. Now, why this is important? This has a lot of medical applications in there, right? So for example, when a cell becomes terminally differentiated, right, it cannot become other cells anymore, right? But this cell still have the same genome. The cells still have the same genome as the cell at the top of the mountain, right? So if we understand the epigenetic information that tells them, now you can divide or you can become that cell, if we know that information, we can probably somehow push that cell at the bottom back to the top, right? Without having to alter the genome, right? Still the same genome, but we can alter the epigenome of that little cell, we can push them back, right? Now, if we push them back and make them so-called pluripotent, now great, right? Let's say, the society is advancing, so everybody's going to get Alzheimer's because we're all going to live to 100 years old, which means, the half of our brain will be empty, right? So what happens back then when we get to that stage? We take a little piece of skin, which represents the little cell at the bottom. We somehow massage it, and it now moves up the mountain, right? Become a pluripotent cell, or become a cell that can now differentiate into neurons. And then we stick them back in our brain. Hey, we can play video games again, right? And the point is, those cells have the identical genetic information, so there's no immune response, no sort of a compromise of, thinking that these are foreign DNAs. There's none of that, because you're not changing the genome, right? You're changing the epigenome. Now, this is going to happen when we know the unknowns, when we are able to chart the unknowns of our epigenome, okay? Epigenomes has played very important role in health and disease, and also in the interaction between our genome, our genetic material, and the environment, right? So I'll give you just a couple of quick examples. When we think about disease, right? So one of the diseases that we study in the lab is cancer, right? Turns out, cancer genome, there are a lot of genetic mutations, so cancer is a genetic disease, but cancer also have a lot of epigenetic changes. So cancer is also an epigenetic disease. Turns out, one of the hallmark of cancer is the DNA methylation change. Cancer cells, globally, their DNA methylation becomes reduced, and locally, their DNA methylation level goes up. As I mentioned, DNA methylation silence genes, right? So one of the key mechanism for cancer cell to kill their tumor suppressor genes, right? For cancer cell to grow, they have to kill the tumor suppressor gene that sort of control the growth. DNA methylation is a very common mechanism that kill those tumor suppressor genes. So that's bad, but that also offers hope. Genetic mutations are difficult to fix. Let's say if we have a tumor suppressor gene where there is a mutation there, it's pretty difficult to go in and change that mutation, right? Epigenetics is reversible. Now, if we have an epigenetic mutation, which is, say, increasing of DNA methylation over a tumor suppressor gene, that tumor suppressor gene is no longer expressed, okay, because you have that big methylation block on it, right? If we come up with a way, somehow, we can remove that methylation, guess what? That tumor suppressor gene is still wild type. There's no genetic mutation there. And then that tumor suppressor gene might be expressed and then be used to kill that cancer cell, right? So that's actually a important venue that scientists are explore that's called epigenetic therapy, right? So we can take advantage of the fact that genetic mutations are difficult to fix, but epigenetic mutations can be reversed, right? So if we can reverse those epigenetic mutations, we may find an alternative way to more efficiently cure cancer cells, okay? So that's with disease. What about environment? I'll give you one sort of a last example before closing the presentation. So, this is a chemical that's been around for many, many years. It's been used in many plastic products. If you have a water bottle, right, make sure you look at it, it's BPA-free, right? BPA is a chemical that's actually found in in 95% of the human beings. So this is an experiment that people did using our lovely agouti mice. So I'm going to bring this example back, right? Turns out chemicals like BPA can shift the phenotype of these animals towards worse phenotype. If you look at the phenotype is a spectrum phenotype. You can be yellow, you can be agouti, and by giving the animals BPA diet, you actually push the phenotype towards more yellow, which is the worst phenotype. They're more cancer prone. So that's a primary example telling people that you ought to watch out for environmental impact on our health by looking at our epigenome, right? So environment certainly has a pretty profound impact on our health through the epigenome. The good thing about the epigenome, again, is it's reversible, right? So it turns out, if you look at different types of environmental exposure, in this case, another vitamin supplement, I feel that I should do a commercial. So, I mean, let's say this is sort of the typical vitamin supplements that people take, and if you give this to the same animals, you're actually pushing the phenotype towards, good, right? So if you look at the pink bars, that's actually more like the wild type phenotype. So so this means that we can actually manipulate, right, through diet, exercise, through, we can't change what our genes are, right? Whatever our parents gave us, that's it, right? So there's nothing we can do, right? But looks like we can manipulate and change our epigenome, therefore change our, phenotype and, potentially fate through diet, exercise, and all these other things, right? So the last experiment, if you give the animals both, right, both the bad thing, the BPA, and the good thing, the vitamin, right, the effect actually cancels out each other, right? So what does this tell me? This is actually very important to me because, I run a lab, I have a pretty busy life, pretty stressful, I have many students I need to work with. I'm very excited working with them, right? I'm very excited about my lab. But, the life is very, very stressful. So, what I do, at the end of the day, I drink, right? So sometimes I have the good things. I grew up in China, but I have the gene that allows me to drink. Sometimes, I drink quite a bit, right? Drinking is probably not that good, right? So, especially to your health, to your epigenome. But I know, as long as the next day I eat some broccoli, I'll be good as new, right? All right, so I'm going to skip this. This is just to show, there's a whole lot more unknowns in our genome, especially even though we know that what our genomes look like, but the large majority of the genome, we still don't know what they do. So there's actually has been a pretty big initiative just over the past few years, from 2009 to last year, to chart the human epigenome, right? Essentially to create the new reference of human epigenome. So we have a reference human genome, as I showed in the printed books, right? So now we just finished an effort, and a large international effort to chart the human epigenome by measuring DNA methylation, histone modification, all these marks that I told you about sort of genome-wide across multiple individuals, across multiple tissue types. So the effort is led by many US scientists. There are multiple mapping centers, Broad Institute, U of Washington, UCSF, UC San Diego. And right in the middle there is our school, Washington University, just down the block, right? So my lab actually played an important role in this project in building a genome browser, right? So in fact, all the data generated by this consortium eventually came to St. Louis and then stored on the 5th floor in our building. And we present this data to the scientists and public all over the world, right? So if you're interested in checking out that URL, Epigenome Gateway. This is why I call, what we do is the gateway to the human epigenome, because we call our browser the Epigenome Gateway browser. So if you're interested, please check it out. So this is the data. So we have profiled across multiple tissues and cell types and individuals, multiple types of chemical modifications that sort of determine how our genomes are expressed, okay? This is what the data look like, and I want to finish by quoting what the President Barack Obama said about the Human Genome Project. That is, back when we mapped the human genome, right? Every dollar we invested to map the human genome, returned $140 to the economy. That was like back in 2013, and then that returns continue coming back, right? So what we hope is the effort that we spent in mapping the human epigenome will have an even bigger return, because we are at the very forefront in charting that unknown. Okay. So with that, I'd like to acknowledge the people who've done most of this work. This is my lab. This is a pretty young and dynamic group. This is in the backyard of my home, and we're having a summer party there. Our lab has been involved in several consortium works, including a project called Roadmap Epigenomics project that gave us this reference map. We have a lot of local, national, and international collaborators. Finally, I want to just put this proposal out there. This is Barbara McClintock, who discovered the transposable element. Now remember, the element that caused the agouti phenotype is a transposable element. So she discovered a transposable element. That's actually the centerpiece of our lab's research. So I propose to put her on a $20 bill. It's a little bit late, but we'll continue pushing it. All right. Thank you very much. Okay.